Okay, I will be presenting problem 15 for chapter 3, section 3.2. In cucumbers, the orange fruit color is dominant R, which means it is big R. And the dominant for the cream fruit color, which is recessive R or a small r. The cucumber plant homozygous, which is big R for orange fruit, is crossed with the plant homozygous for cream fruit, which would be small r. The F1 are intercrossed, F1 individuals crossed with F1 individuals to produce F2. So the first part of our problem is asking, give the genotypes and phenotypes of the parents, the F1 and the F2. So the parent was already determined for us here, and the parent would be homozygous dominant orange fruit, which is big R, and homozygous recessive cream fruit, which is small r. Get that? So the Punnett square is going to be big R across the top and small r down the bottom, and that up and down. And then we would cross those to get our F1, and the F1 would be 100% heterozygous, big R, little r. It's still going to be orange because it has that dominant R in there. So the phenotype for F1 will be orange. Type is big R, little r, and it is 100%. Then we're going to cross that to get our F2. Little r across the top, big R, little r going up and down. And we're going to get one homozygous dominant, big R, and that's going to be 25%. Then we're going to get two heterozygous, little r, that's going to be 50%. But since it still has that dominant R, it's still going to be a phenotype of orange. Have one homozygous R, which will be a cream. So we have 3 orange, 1 cream. 75% orange, 25% cream. The next part of our problem asks, give the genotypes and phenotypes of the offspring of a back cross between the F1 and the orange fruit parent. Remember from our previous problem, the F1 is going to be big R, little r, and that phenotype is orange. And then we're back crossing it with the orange fruit parent. Parent is homozygous and is dominant, big R. Have big R, little r across the top, big R going up and down. And that's going to give us one big R, which is homozygous dominant, and one big R, little r, which is heterozygous. But the phenotype is going to be 100% orange phenotype because we have that dominant R crossed. Problem, it asks, give the genotypes and phenotypes of the back cross between the F1 parent and the cream fruit parent. So the F1 parent remains the same. We have big R, little r, heterozygous. But now we're crossing it with the cream fruit parent, which is homozygous recessive, small r. Big R, little r across the top, small r going up and down. And it give us one big R, little r, heterozygous, one small r, or 1/2 orange and 1/2 cream. And that is how we use the Punnett square to understand the genotypes and the phenotypes. And the Punnett square looks literally like a square, E drawn in the middle.